In this video, we'll discuss a new method for early cataract diagnosis and explore potential non-surgical treatments. We'll also explore the broader potential applications of this method, as the mechanisms that cause cataracts also underlie Alzheimer's, Parkinson's, and other protein misfolding diseases. In this context, the eye's lens represents a powerful model for developing and testing potential drugs for these conditions. A cataract is a clouding and discoloration of the lens of the eye caused by increased scattering and absorption of light. Most cases of cataracts occur in older people, and 1 in 2 people over 80 in developed countries has undergone cataract surgery or is awaiting it. Preventive cataract treatment cannot be effective without early diagnosis. If treatment is started at the earliest stage of cataract development, while structural changes in the lens are still reversible, the chances of success are high. The primary function of the lens is to focus light onto the retina. The lens consists of an ordered arrangement of differentiated fiber cells that have lost cell organelles and nucleus. This makes protein turnover impossible, and once synthesized proteins remain in the lens for life. To maintain metabolism, these cells are interconnected by so-called gap junctions, which ensure the circulation of ions and antioxidants and the removal of waste products. The high refractive index of the lens, 1.38 in the cortex and 1.41 in the nucleus, is due to a very high concentration of protein increasing from 200 to 400 milligrams per milliliter towards the nucleus. Despite the high concentration, proteins remain soluble, providing high homogeneity to the refractive index. However, modification of the protein may lead to precipitation, resulting in an increase in light scattering. Since one eye lens protein contains an average of 4 tryptophan residues that absorb UV light, the lens has an extremely high optical density in the UVB range, forming a long-pass filter which protects the eye and the lens itself from toxic UV radiation. The young lens is transparent in the visible range up to the near UV. However, over time, absorption in the visible spectral range builds up. We and others showed that this absorption results from the formation of post-translational modifications in the lens proteins. This figure shows the absorption spectra of the most abundant fluorescent post-translational modifications within the visible range. Notably, many of these post-translational modifications are the result of photo-oxidation of the tryptophan side chain. This is, however, a rather paradoxical finding, since tryptophan in the lens cannot be excited by UV light due to the long-pass filter caused by its absorption. The optical density of tryptophan absorbance is so high that UV light can get in just for a few microns into the lens. Therefore, the question arises of how the lens tryptophans are getting excited to form the photoadducts. To answer this question, we need to take a closer look at tryptophan spectroscopy. We have previously shown that the electronic spectra of the tryptophan side chain depend on the polarity of the microenvironment. The figure shows the excitation and emission spectra of the same dye dissolved in low, medium, and high polarity solvents. The higher the polarity of the solvent, the redder is the wavelength of the spectral band. The spectral shift of tryptophan between non-polar solvents, such as cyclohexane, and highly polar solvents, such as water, is about 42 nanometers, which is comparable with the half-width of the spectral band. An emission spectrum of a multi-tryptophan protein is a superposition of the spectra of its individual tryptophan residues, which are located in different polarity environments. If the side chain is buried in a protein body where polarity varies from low to medium, The absorption spectrum has a maximum at 280 nanometers, and there is no absorption beyond 310 nanometers. But if a side chain is on the surface of the protein and therefore is exposed to the aqueous environment, the absorption spectrum shifts to red, and then this fraction can be excited selectively in the 310 to 320 nanometers range. This water-exposed red fraction is strongly susceptible to oxidation by reactive oxygen species. Therefore, we suggested that it is this fraction that can be responsible for triggering the process of photo-oxidation in the lens, since it can be excited by UV light inside the lens and possesses an increased reactivity. This red-shifted tryptophan fraction acts as the vanguard, breaching the lens's natural UV defenses. Once this protective barrier is breached, visible light takes over, oxidizing the visible absorbing tryptophan adducts and triggering irreversible structural damage within the lens. To make matters worse, certain tryptophan adducts, such as N4-mylkynurenine, are powerful generators of singlet oxygen, further accelerating the oxidative process. Concentrations of fluorescent adducts can be semi-quantitatively determined from decomposition of the emission spectrum of the eye lens. We found that the total concentration of the first three of them correlated well with the grade of cataract. Thus, the method provides accurate and sensitive cataract diagnosis at the molecular level that is far superior in sensitivity to light-scattering methods such as the slit lamp or optical coherence tomography. Its main advantage is that it allows diagnosing the latent phase of cataract formation until the moment of irreversible damage to the lens. The concentration of fluorescent post-translational modifications can be measured at micron resolution across the lens using confocal fluorescence microscopy. The turquoise fluorescence in panel C reflects induced emission, the intensity of which is directly proportional to the concentration of post-translational modifications in the lens. Simply put, the brighter the glow, the more changes the protein has undergone. Cataracts are a case study in protein misfolding. The proteins in the eye lens never turn over, so over a lifetime they accumulate modifications and damage. This leads to misfolding and aggregation, which eventually build up lens optical density and opacity. The eye lens offers a unique research window, as fluorescence techniques can reveal early molecular changes well before any visible structural damage emerges. The eye lens is unique because it lets us observe protein misfolding directly, yet cataracts are only one member of a much larger family of protein misfolding diseases. Similar misfolding-driven damage occurs in Alzheimer's and Parkinson's disease, and even in some cardiomyopathies, where alpha-crystallin B plays a chaperone role. In conclusion, early cataract diagnostics can detect cataractogenesis at a very early stage and support the development of non-surgical therapies to prolong the lifespan of the eye lens. Moreover, the lens is a powerful research tool, a real tissue system where we can watch proteins misfold, track how drug molecules penetrate and are retained, and quantify the structural changes they induce. The lens is not only a therapeutic target, it is a unique in vitro model for protein misfolding research. Insights from cataracts can help guide therapies for other protein misfolding diseases.